Hello, my name is Francisca Sanchez. Today is July 17th, 2016. It is week 3, lab 3 of Bio 225. Today I'm going to be doing the microscopy for microbiology, and I'm going to be demonstrating the letter E to focus it on. The letter E using the 4 different lenses, and then I'm gonna do the wet mount slides as well. I'm gonna do my cheek swab. So first what I did was I wiped down the lens and the slide with the lens paper, and this was already before I wiped it down. I measured it with my ruler. I focused and measured it, and then I put the letter E slide. It's in there already. I don't know if you can see it, but sorry. The letter E slide is in there, and I'm going to focus it using the scanning lens first. And right now I've already have it focused. I've taken my photo of it. So now I'm going to change from the scanning lens to the medium, which the scanning lens is the 4X, and now I'm going to go to the medium lens, which is the 10X. So I'm going to just change it. And I'm going to try and focus it. Okay. So I focused it. Sorry, I should have explained. With the scanning lens, the E was very visible and clear. With the medium one, it actually enlarged the letter E. You can still see the letter E. It's not quite as clear, but it's still clear. You can make out that it's a letter E. Next one, I'm gonna drop the table a little. Or actually, sorry, I'm going to raise my lens. And I'm going to switch it now to the high, which is the 40X. This is hard to switch it. Okay. And then I'm going to bring it back down into focus. And so when it's on high, it's magnified the letter E, so I can only see part of it, and you can see it's gotten blurry as well. So that those are the 3 lenses. Now I'm gonna take off the scanning lens. Sorry, I don't know if you can see that. And I'm going to add the oil immersion lens. So I'll place that in there for now. And we're going to do it. Now we're going to look at it through the oil. And the oil immersion is 100X. Okay, so it is asking that I apply a drop of immersion oil on the specimen slide and turn on the nose piece to bring the 100X lens into position. So I'm going to take my lens, and it wants me to apply a drop of immersion oil on the specimen slide and turn the revolving nose piece to bring. Okay, so I'm going to drop immersion oil. Okay, we applied a drop. Okay, so now we're gonna put the slide back in, holding it. And we're going to, with the lens tip touching the oil, focus with the fine adjustment knobs. Okay, so I'm going to bring this back into focus. And I'm going to be touching. Oh, I need to turn my light back on. Okay. So this one. Sorry. Okay, I'm gonna make sure it's touching. And I'm not sure. I'm finding it. Sorry. Oh, and okay. Okay. Oops. Having a hard time finding it now. It's touching the slide. I'm trying to bring the focus in. And I'm not exactly sure why I can't see it. But maybe I did something wrong. Okay. Alright, hold on. Okay, so I finally got it into focus, and you can see the entire letter E, but the whole thing is blurred. So now I'm going to remove my slide. I'm going to clean it off and replace it back into my little box of slides, clean it up, and then I'll come back and do my wet mount cheek swab. Okay, I'm back and I'm going to do the wet mount slides. So the first thing I'm going to do is I've gathered all my materials. They're all right there. Distilled water. Okay. And I am going to place a sheet of paper onto my workspace to prevent the slides from cover slips from getting scratched. I'm going to put on my gloves and my safety goggles. Okay. Okay, I'm going to gather a clean slide and the cover slips. So, clean slide, cover slip. Place them on a paper towel. I'm going to use one sterile swab from the package. Gently scrape the few cells from the inside of my cheek. Okay, so, okay. Smear the cotton swab in the cheek. Smear the cotton swab containing the cheek cells onto the microscope slide. Carefully add a drop of this cell. I'm going to put it here. Okay. Carefully add a drop of distilled water. A raindrop. Okay. And then I'm going to add, holding the slip upright, I'm going to gently apply the slip and allowing the water to make contact with the slide, the slip, the cover slip, before actually placing it on there. So upright, place at the edge, and there it made contact. So now I can lay it down. I can use the napkin to absorb any water, any excessive water on the sides of it. Just putting it on the edge to absorb water. Okay. Okay, now I'm going to place it on, I'm absorbing all the excessive water. Now I'm going to place it onto my stage, microscope stage. And I'm going to focus the cells under scanning power. And then refer to figure 15 when locating the cells on the slide. Okay, so I'm going to do it now. I have the scanning power lens, this one, the 4X. I'm going to focus on it, see if I can see some cheek cells. So they should look like, it says to refer to this picture, so it should look similar to that. Okay. Alright, just going, okay, lowered it, coming into focus, and I see something. Oh yeah, it's similar to that. So I focused in on that cheek cells. So it says switch the objective to lower power and focus on the same cells. Switch it to the higher power and focus on. So now I'm going to just switch it from the lower power to the higher power. And I'm going to take a photo of all those as I switch them. I'm going to so once I get the pictures of those, I'm going to do the neck swabbing and do my teeth. But I need to take photos of those first. So there's 2 parts of this lab that I have completed. Thank you.